Hello everyone, this is Madhavi and I am talking about the search for genetic material. As everybody know that there are two types of genetic material, that is DNA and RNA, right? And among this only they wanted to tell us that whether DNA is a genetic material or RNA is genetic material. Why? Because of these two are present in almost all the living organisms, but viruses are the ones, some of them are having the RNA as genetic material and which causes plant diseases, and some of them having the DNA as genetic material, mostly they will cause the animal disease, except retroviruses and also cauliflower mosaic viruses. They are exceptional with this, but still they wanted to prove that. Here Sutton and Boveri also try to tell us that about the chromosomes are the carriers of the genes also. Later T.H. Morgan proved that genes are located on the chromosomes present in the nucleus, but the molecular nature nobody has been detailedly explained. So here we will discuss about some of these experiments. Among the first one is transforming principle. Transforming, in the name itself it is a something is transformed from one organism to another, so that the another host is going to change its properties. That's why we call it transforming principle, and it is the most famous also. And bad luck is no, some scientists did not satisfied with these experiments, and also later whoever proved this, right, we will talk about that also. Now, Frederick Griffith in the year 1928 performed a series of experiments by selecting the two strains of the bacterium, that is smooth strain and also rough strain, which are present in the Streptococcus pneumonia, which is also called a pneumococcus, and causing the pneumonia disease, right? Now, S3 strain is a smooth or capsulated type, have a mucus or polysaccharide coat also, because it produces smooth and shiny colonies in the culture plate. So, and are also virulent. Virulent means causing the disease, causing the pneumonia also. Now here when you talk about this R strain, that is rough or non-capsulated type, have no mucus coat, have produced rough colonies also. These are non-virulent and do not cause the pneumonia. So the series of experiments will be followed like this. The first thing is that they have taken the smooth strain and they have injected into the mice. Later mice die, because we know that smooth strain is causing the disease. Now R strain also they have taken and they injected into the mice, and mice was live, because R strain do not cause the diseases because of its rough nature and do not have that capability. Next what they have done is S strain, they have heated it, so we call it heat killed it, and everybody will think that this is the one which is having the DNA structure, but it got died. So injected into the mice, mice was live, because it did not, after eating this nature only, renaturation and denaturation are the two properties will be happening, ma. So that's the reason mice was live. Next smooth strain only heat kill plus R strain also, both of them do not cause a disease, that is why injected into the mice, but surprisingly mice was died. What was the reason for this? Everybody shocked, because Frederick Griffith conducted, in that situation he was also shocked. So he was tried to explain that it was a transforming principle, who is transforming from S strains to, but he did not explain the detailed description about the whether the DNA is genetic material or what is that. Later on the transforming principle been again reconducted, again proposed by the Oswald Avery and Cohen and Magliard and Maclean McCarthy in the year 1933 to 34. So here it is conducted in the 1928. Later on 1933 to 34 they conducted an experiment, again rediscovered this experiment. What was that experiment? This experiment, for some people will not understand, you try to better understand with this language. Heat killed S cells. That means what? S cells, whatever the DNA properties DNA is there, it is present, but it doesn't have the ability to cause a disease now. Okay, now it is called as a heat killed S cells. From this only they have taken in the test tube, and later on they have removed the lipids and carbohydrates, because we don't want the lipids and carbohydrates which are present inside the bacterium and other things also, right? They don't want, they removed this. So removal of lipids and carbohydrates has been done. Later on this whatever removed no lipids and carbohydrates, then they split this material into 3 test tubes. In that first test tube what they have added is, they have added the proteinases actually. What is the function of proteinases? See, everybody know that in every cell there is a presence of lipids also, carbohydrates also, proteins also, DNA and RNA and other materials also present, right? Now here what they have removed, lipids and carbohydrates removed. What is remaining then? Proteins are remains, DNA is remains and also RNA remains. So they wanted to prove that whether proteins are carrying and changing the properties of our cells. Why the mice was died in the last experiment, it wasn't their doubt. So later on they wanted to prove that. For this only they have added proteinases. These are the enzymes. So this is an enzyme which kill the which break the bonds between the proteins. So when they added proteinases to these proteins, then protein bond was changed. So the proteins do not have the energy or nothing to transform, no. Then in the another test tube they added ribonucleases. Ribonucleases means they are the ones which are breaking the bonds between the RNA. Next, they added deoxyribonucleases, and these are the ones which break bonds between the DNA. That means here proteins are absent. That means what are present? DNA and RNA are present here. Next, here they added ribonucleases. That means RNA was not present. Then remaining what are present? It is proteins and DNA is present. Now here DNA they have removed. That means DNA was not there. What is present then? Here it contains the proteins and also RNA. So these are the three test tubes which they have conducted. Later on they have taken into the flask. This what is the material? This material has been taken into the flask. Now this contains only DNA and RNA, but not proteins, but not lipids and carbohydrates. Then for this only they added R cells. R cells means again rough cells. So here already S cells are there with the DNA and RNA, and also R cells they have added. Then what happened? S cells are appearing again. Already we have seen that heat killed S cells. S cells are not having a live only, but here live S cells have started appearing. That is the reason mice was there, no? So that's the reason, right? Because mice was died. Why it got died? Because DNA and RNA are present, no? Among these two, any one of them has transformed the S cells into R cells. So R cells and S cells have transformed and mice was died. Maybe this was the reason they thought it. Okay, later again. Here they have added R cells. Now what is present here? Proteins and DNA is present, no? Again the S cells are appear. Again the mice was died. Again they got confused. Hare, here proteins are not there, but DNA and RNA is present. Maybe we are conducting that for RNA and DNA, but here proteins are present, DNA is also present. Maybe something is causing the disease and mice was died. Later they added R cells to this, which contains only proteins and which do not contain the DNA now, which contains proteins and RNA. Now no S cells are appear. That means mice was live. Why? They come to know now. Because when DNA is not present, R cells are not transformed to S cells. That means proteins and RNA are not transforming from S cells to the R cells. That means nothing will happen. Mice was live happily. That means they confirmed that DNA is the genetic material which is transforming from S cells to R cells, so that S type of cells only are appearing. But after this much of conduction also, still some of the scientists are not satisfied. So that is the reason many of the experiments have been conducted. In the next class, we will be knowing about all this. Please subscribe my channel, share it. Thank you.